This is a video on the two types of cell division, mitosis and meiosis, presented by Simon Bailey, Hunter Hatzenbeeler, Bryson Ingebretson, and Cale Ingebretson.
For interphase, mitosis is the process of cellular division. However, before mitosis can start, the cell must go through an interphase stage. Interphase is broken into three parts: growth phase 1, synthesis, and growth phase 2.
During the G1 or growth phase 1 stage, the cell does its job and performs its normal functions. It also begins to grow. This is the longest period of interphase. This is a picture of what a cell looks like during growth phase 1 of interphase. Notice it still has the nucleus, nucleolus, and centriole. Right before the cell is ready to enter the synthesis stage, it enters a period of rest called the G0 phase. This is where it rests and gathers all of the energy needed for DNA synthesis. The second part of interphase is synthesis. In this stage, the cell replicates its DNA. Although the cell continues to grow, the emphasis of energy usage is for DNA replication. This process requires a large amount of energy, and at the end, the cell will have an identical copy of its DNA. Here's a picture of DNA replication when a DNA strand is copied and turned into a chromatid.
The last part of interphase is the G2 or growth phase 2. The cell will continue to grow, but the most important energy usage is for the replication of all the cell's organelles. Also, the microtubules will begin to organize and form the spindle fibers. These spindle fibers will be used later in mitosis to form the spindle apparatus. Here's a picture of what a cell looks like at the end of the G2 phase of interphase.
And that is the end of interphase. After interphase, which is the longest period in the cell cycle, the cell begins mitosis. Mitosis is the process of division in the cell. Its purpose is for growth, development, or repair. The first stage of mitosis is prophase. In prophase, the chromatin condenses and thickens into chromosomes. This protects the DNA as it is being transported throughout the cell. Then the nuclear membrane dissolves. The nucleolus also dissolves after this. The asters, which are made up of a centriole and spindle fibers, move to the opposite poles of the cell. As they are moving, the spindle fibers stretch out across the interior of the cell and form the spindle apparatus. This helps provide structure to the cell as it goes through mitosis. Here is a picture of what a cell would look like during prophase. Notice the asters on both sides. The second stage of mitosis is metaphase. Prometaphase. In this stage, a second set of spindle fibers attach from the asters at the poles of the cell to the kinetochores of each of the chromosomes. Once they are attached, congression occurs. Congression is the push and pull movement of the asters as they move the chromosomes close to the center of the cell. This is a picture of a cell during the prometaphase stage.
The third stage of mitosis is metaphase. In this stage, the alignment of the chromosomes is adjusted in small movements. Metaphase is complete when the centromeres of the chromosomes is lined up perfectly on the metaphase plate, which is an imaginary plane that splits the cell perfectly in half. This is a cell during metaphase. Notice how the chromosomes are lined up perfectly in the center.
The fourth phase is anaphase. There are two parts to anaphase that happen at the same time to help the cell to start separating. The first part of anaphase is anaphase A. In anaphase A, the spindle fibers connected to the chromatin begin to slowly dissolve, pulling the chromosomes towards the poles. In anaphase B, which happens at the same time, the membrane begins to pull backwards with the aster, which spindle fibers are connected to the chromosomes, and they all pull back, creating a peanut-like shell, as seen in the picture down here, which is a cell during anaphase. Notice how the chromosomes are separated perfectly in half, so there's the same amount of chromosomes on each side. The fifth and final stage of mitosis is telophase. Telophase is basically the opposite of prophase. First, the nuclear membrane returns. Then the nucleolus returns. After that, the spindle apparatus dissolves. And then the chromosomes relax and unwind back into chromatin. Finally, the organelles that were copied in interphase are organized and separated so that there's exactly half of each of the organelles on each side of the cell. Again, this is a picture of a cell during telophase, and you can see that the organelles have all been organized and separated.
Here is a short animation of a cell going through mitosis from prophase to anaphase. Throughout the process of interphase and mitosis, there are 3 checkpoints. These checkpoints check for accuracy and allow the cell to respond to both external and internal signals. There's a checkpoint in between the G1 and S phase, between the G2 and M phase, and during the late metaphase.
The one checkpoint we are going to focus on is the G1 and S checkpoint. This occurs between growth phase 1 and synthesis phases of interphase. This checkpoint checks to see if the cell is healthy enough and has enough energy to divide. It takes into account if the body is sick and if it needs to use the energy elsewhere. If the checkpoint is missed, like if the cell doesn't have enough energy to go on, then it cannot divide. The energy is then used somewhere else in the cell, and the process of mitosis will not happen. Meiosis. The main difference between mitosis and meiosis is their purpose. The purpose of mitosis is to divide the cell so that it can grow, develop, or repair. The purpose of meiosis is to create haploid gametes, which are cells with only half of the cells in their chromosomes in their DNA. These cells are egg and sperm cells in humans. Here's a picture showing the contrast between mitosis and meiosis in different cells.
Meiosis is very similar to mitosis, but there are some definite differences. For the purpose of time, we are only going to go over different parts of meiosis rather than every phase. Meiosis is split into 2 stages, meiosis 1 and meiosis 2. To start, one major difference in meiosis is in the prophase stage. In meiosis, synapsis happens, which is when the chromosome pair from each of your mom and dad are matched and stacked on top of each other. They are glued together with the cohesion protein. This is so that later in the process of division, each chromosome is separated rather than individual chromatid. The synaptonomal complex refers to all of the chromosome pairs, or just one of the pairs, being glued together during synapsis.
Another process that happens in meiosis and not in mitosis is genetic recombination. Genetic recombination happens in prophase 1 of meiosis. When the chromosomes are paired, they cross over between each other. When they twist together, DNA is shared. This process is called genetic recombination. One point where the chromosomes cross over is called a chiasma, and multiple points where the chromosomes cross over is called chiasmata. Genetic recombination is important because it is the foundation for evolution. This DNA sharing allows offspring to be better than both their parents since they get both of their traits. And here's a picture showing the entire meiosis process. The final result of meiosis as compared to mitosis is seen in this next slide. At the end of mitosis, the cell has created 2 genetically identical daughter cells. These cells are diploid and have all 46 chromosomes. They are identical because they never underwent genetic recombination and were divided perfectly in half during anaphase, making them the exact same. At the end of meiosis, the cells created 4 genetically unique daughter cells. These cells are haploid, and they only have half the number of chromosomes, which is 23. There is 4 because after meiosis 1, the cell splits, and after meiosis 2, the cell splits again, creating 4 cells. These cells are genetically unique because they undergo genetic recombination.
Here is a picture showing a comparison between the results of meiosis and mitosis. Notice the amount of daughter cells and the amount of chromosomes in each cell.
And that is the end of our presentation on mitosis and meiosis. Thanks for watching.